Alright, today we're going to take a look at the examples from the Beyond Mendel worksheet. This is looking specifically at incomplete dominance, codominance, multiple alleles, and polygenic inheritance. You can write this in your notebook. There should be a space for a box where you can put the definition for incomplete dominance, and then I'll do one of the example problems from your paper. Whenever we're looking at incomplete dominance, we're really just looking at a different way to interpret what the Punnett square shows us. And so with incomplete dominance, this is a trait that is going to be inherited, and it's going to be a trait that means that both of the alleles have equal, like one is not dominant over the other. So this means that whenever we have a heterozygous individual, then the phenotype is going to be a blend of both of the alleles. So we won't see either of the alleles, we'll see just some blend of it. So we're going to do number 1 as the example for this. And so in number 1, and then also number 2, but in number 1, the red 4 o'clock flowers have the genotype R, so homozygous dominant. White 4 o'clock flowers have the genotype r, and then the heterozygous flowers appear to be pink. And so whenever we're talking about this, in this problem we have two alleles for a certain trait. We have R, which is red, and we have r, which is white. Whenever we're looking at these, though, they create different phenotypes. So, R will be red, lowercase r will be white, and then whenever it's heterozygous, now it's going to be pink, which is a blend of that red and white trait. These, I think, are actually a little bit easier to pick out because the heterozygous is different, so you don't have to determine, oh, it's dominant, is it homozygous dominant or heterozygous? Now we know because there's 3 different phenotypes that we can look for. All right, so for number 1, it says, what are the probable genotypes and phenotypes of the F1 generation when both parents are pink 4 o'clock flowering plants? So, whenever we're looking at this, we know that we're going to cross our parents, and the parents are going to be, parents are pink. So, if the parents are pink, then we know that they're heterozygous, so R, lowercase r, and then we want to write down and find the genotypes and phenotypes. So the geno and pheno of the offspring. Okay, so we're going to do our Punnett square like we always have. Our Punnett square then will show us that we have here, so R, lowercase, R, lowercase. So whenever we're looking at this, the genotypes, the probability for our genotypes would be the same as it is normally. 1 homozygous dominant, 2 heterozygous, and then 1 homozygous recessive. Okay, our phenotypes is where it's different. So our phenotype ratio, we would say that we have 1 red to 2 pink to 1 white. And so instead of having that 3-to-1 like we saw in the dominant and recessive inheritance, now we have a 1 to 2-to-1 ratio for the phenotypes as well. Okay, so you can give the other incomplete dominance problems a try, and you can check them off with me whenever you're finished. Let me know if you have questions. The next type of inheritance we'll look at is codominance. So people like to get this one confused with incomplete dominance. Think of incomplete as nothing is better, so there's a blend, but codominance means that both traits are equally dominant with one another, and so if you have a heterozygous individual, then you'll see both alleles in the phenotype. We're going to do practice problem number 5. Number 5 is looking at chickens, and so feather color in chickens sometimes is codominant, and the feather color can be represented as black, so it can be black color, which would be C capital B, C capital B. It can be white, which would be CW, CW, or it could be erminette, which is essentially just a mix of both of these, and where you see both of them. So you would see black and white feathers. So this would be CB and CW. Okay, we can use the different letters because both of these traits are equally dominant, so one isn't over the other, and then whenever they're together, we see both black and white. All right, so we'll do number 5. We're saying that we're crossing a black chicken with an erminette chicken. And so we'll go ahead and write down their genotypes. So C capital B, C capital B, and then C capital B, C W. All right, so we'll do our Punnett square. And the question asks, what are the expected genotype and phenotype ratios? So what are the ratios? All right, so we'll put the black chicken at the top, the erminette chicken along the side, and then we'll go ahead and fill this in. Oops. If you want, you could shorten this to just put BB and use just the B and W. That would be fine too. This is just as a reminder. Using the C's, I think, helps you to remember that it's codominant, so you see both black and white. Okay, so we're going to go ahead and analyze our genotypes and phenotypes. And for our genotypes and phenotype ratios, they're going to be the same. So our top yellow part, and then we'll do the bottom, our green. These are offspring are going to be the same. So we'll say for genotype ratio, so for our genotype, let me just erase that. Okay, so for our genotype ratio, we're going to have 1 CB, CB to every 1 CB, CW. Okay, the same will be true as the phenotype, except that we'll say what those physical characteristics are. So we'll have 1 black chicken for every 1 erminette chicken for our potential offspring for this cross. You can go ahead and take a look at 6, 7, 8, and 9. Let me know if you have any questions, and we can go over it in class. Okay, so after you finish the codominance problems, we'll take a look at our multiple allele problems. And this is going to be a trait or some traits that have more than 2 alleles. So that's not just that you have a red allele and a white allele. Maybe you have a red and a purple and a green allele for a certain trait. And it's going to code for 3 or more phenotypes. Remember that each person still only gets 2 alleles for a certain trait or for this trait. It's just that there are more possibilities than just the 2. One of the most common things that we look at with multiple alleles is your blood type. And so blood type is, there's multiple alleles. So you have an A, a B, and an O. And whenever you have A and B, they are codominant to one another, meaning if you have both of them, you see both A and B in the phenotype. So the tables that are listed on your worksheet, you should have got this again in class, or this is also on Blackboard if you need it. But it shows tables for what the alleles are. So there's IA, so our alleles for blood type. So we're going to do number 11 together. But IA is A blood type, or A type. IB is B type. And then little I represents O type blood. This is really the recessive allele, so it only is expressed if you have 2 of them, and then these 2 are codominant, so if you have 2 of them, then they're both expressed. Okay, and then it shows the different combinations of how you can get different genotypes and phenotypes. All right, so in number 11, it says, what genotypic blood types must the parents have in order to produce offspring of blood types A and O? So what are the parents? If the kids are type A and type O. All right, so type A. Type A, whenever it is going to be type A blood, we know that that genotype can be IA, and then this second allele is unsure. We just know that it can't be IB. Type O blood has to be little I, which is essentially the recessive allele for blood type. All right, so we're going to do like we have in the past where we've worked backwards. So this is these ones that we put up here. These are the offspring, and we want to find the parents. And so they have 2 alleles for these traits. We know that based on the O blood type that each parent has to at least have the I, the little I, and then at least one of the parents has to have I capital A. So this missing spot can either be IA or I. So we're going to just do 2 Punnett squares. So we're going to have IA and I, and then we'll just say that this one's IA, I, and then we'll do a second Punnett square that's going to look at IA, I, and then I. Alright, so if we fill it in, I'm going to try to fit this in here. So if we fill this in, then what we should find is that actually both of these are possible because they only produce I and O phenotypes for blood. So you could include either one. Both are correct answers for this, which is different than what we've looked at with some of our other Punnett squares. So you can go ahead and take a look at the other multiple allele problems and check them off with me when you're done. Ask me any questions that you have and we'll go over them in class. All right, the last type of inheritance that we will look at is we'll look at polygenic inheritance. And a good example of polygenic inheritance, polygenic traits. And so polygenic traits, that's a trait that's going to be controlled by 2 or more genes or multiple genes that are on different chromosomes. So there's multiple genes that contribute to one trait or one phenotype that we see. So the example of this that we'll use is coat color on labs. And there's 2 genes that we look at that are responsible for their coat color or for their pigment. And so you can write this in your box in your notes, but color in dogs is controlled by one gene where we have an allele for black color, and then we have an allele for brown color. Okay. There's also a gene then that is going to be for the pigment deposition, so the pigment deposit. And this one is going to be, we'll use capital E for the pigment is deposited, is seen or is there, and then lowercase e for no deposit, meaning that you won't see any pigment being actually put into the fur color. So both of these together contribute to the overall color of dogs. So for the dogs, we can have a black lab, we can have a brown lab, and we can have a yellow lab. This is going to be, because I didn't mention this is number 19, so you won't have to do 19 again in your example paper. So for the black color, for any of the possible genotypes for black, you have to have at least the capital B allele, and then it can be capital or lowercase, and then you also have to have the capital E, and it can be capital or lowercase. So it has to be dominant, must be dominant for both. Okay. For the brown, you have to have lowercase because it's the recessive trait, and then you have to have the capital E. So you need to be able to have the recessive and then the capital E in order for the pigment to actually be deposited so that you can see it. For the yellow lab, it doesn't matter, so this doesn't matter. The color doesn't matter what it is. It can be dominant, it can be recessive, it can be heterozygous, it doesn't matter. The only thing that matters is that this has to be recessive. So it's recessive for pigment, for the depositing of pigment, which means that it doesn't matter what the color is. So the color doesn't matter, and it can be anything. Whenever we're looking at polygenic inheritance and polygenic traits, it's just the interaction between those 2 genes to see what the phenotype is that are produced. For 20 and 21, you can use this information that we've written down here and take a look at the phenotypes to figure out some of what the genotypes are for the parents. It won't really require Punnett squares, just some reasoning on your part as to how inheritance works and how that recessive and dominant trait fits together. So finish those up, ask me any questions that you have, and you can check these off with me when you're done.